the most important fact about her is that she's not from Queensland, practically. I apologize on her behalf, but she will be. Well, Laura comes from Wollongong, where she did her bachelor's and honors, and did a really excellent honor project that she's going to introduce now. That is also very relevant to her PhD. In between her honors and doing her PhD here with myself and Bauerella, she has been working as a consultant, as an ecologist, working with koalas, but she claims that she likes more bees than koalas, and that is what she's here. So those are the most relevant facts, I think. She does have quite a lot of experience on different types of consulting ecology jobs, but decided to go for a PhD, and we're really glad we can have her. And she's already making extraordinary progress on her PhD, and is already producing data. Soon we'll have data in Excel, and that will be very exciting. And she's going to introduce a little bit, just one slide about her PhD, and then jump to her work about her honors. That is very, very interesting and also very relevant to her PhD. So, without further ado, Laura.
Thanks, Daniel. Yeah, hey everybody, thanks for turning up. Now, like Daniel said, my PhD here with Daniel and Laurie is looking at invasion ecology. So the centres that you can see pictured, they've got really specialist interactions in their native range, and yet globally and across the tropics, they're highly invasive species. So sicklepods on the left, which I'm sure everybody in this room knows of. So I'm going to try and look at what factors are contributing to its invasion success when it's got all of these processes that should be acting against it. And the purpose of this slide, before I jump into my honours research, is another little reminder of anybody who's tuning in. If you want to volunteer, please email me, because I need people who want to come and look at bees that are going to land on flowers. And if anybody knows of sicklepod stands, please email me, or any other centre species, because I'm still trying to find some populations. But my honours research, like Daniel said, this was done at UOW, and it was a collaboration between the University of Wollongong and the Australian Plant Bank at the Botanic Gardens. And my project was looking at what factors are contributing to viable seed production in some of the rainforest Rutaceae species we have at home. So what I'm sure is not a shock to anybody in this room, rainforest ecosystems are having huge impacts from land clearing, and it's reducing both the size of the ecosystem and the biodiversity within them. And what may be a bit more of a shock is that we do have rainforests on the south coast of New South Wales. They're small pockets, they've been extensively cleared, and we've reached a point where they're threatened ecological communities. So the persistence of them does rely on conservation efforts and restoration programs. So you can see on this map here, the black pockets, all these little black patches, that's the rainforest that still exists, whilst all the hatched labelled as the Illawarra brush is where that rainforest community was once continuous along that coastal ecosystem. And we're just south of Sydney here in the northern part of the south coast. I would like to point out though that this reference is from the mid-90s, so nearly 30 years old, and their patches are much more fragmented than that at the minute. So in order to conserve these ecosystems, there's an ex situ conservation technique, seed banking, where you physically preserve or store seed. And this is used then, so you can have germination experiments, reintroduce those species and restore those ecosystems, hopefully. But the success of a seed bank will hinge on sourcing viable seed for populations. So a viable seed is going to be filled, contains both the embryo and the endosperm. It's live, it's mature, it's going to germinate when you put it within those right conditions. And that wants to click. There's a number of factors that influence viable seed production. So firstly, plants need to produce a lot of compatible or viable pollen, which is going to be transferred from the anther onto the stigma. So you have different pollination vectors that are going to provide this service. Abiotic processes might be wind or water, and what's quite common in rainforest ecosystems is that it's insect species, so those biotic pollination vectors. But we also want to know whether or not a plant actually requires that pollination vector to set its seed. So some plants will be self-compatible, they might not need a pollination vector, they might be self-compatible and still require a pollination vector, or they may not be self-compatible at all, and then they definitely need something to transfer that pollen for them. And if all of these processes go to plan and you do get a fruit set, there's other factors that might still inhibit a viable seed being produced. So predation, you've got predispersal seed predators, frugivores that might damage the seed and in turn it won't germinate, or other environmental factors. So if you're in a drought condition or the plants are stressed for lacking nutrients, they might abort that fruit or seed. So I tried to look at a broad spectrum of these to build up an idea of some of the factors that are contributing to viable seed production. So then why study the Rutaceae family? Rutaceae are a family of plants. They're common within Australian forest communities. They're quite abundant within the rainforest ecosystems on the south coast. And seed viability is known to be really variable in them. So this source here from the 2009 paper that Martin produced, that was a paper that came out of the plant bank. So when they were trying to collect seed, they found in the Rutaceae plants, depending on the season, fruit set would be really variable. When a fruit was produced, whether or not it contained a seed or several seeds was quite variable. And then when they did find a seed, whether or not that was viable and contained that embryo and endosperm was again really variable. So they were getting huge variation, and often it was quite poor within the rainforest Rutaceae species. So my project aimed to look at some of these factors that might be contributing to this huge variation in seed set for rainforest Rutaceae. And to achieve this, I had 3 subsequent aims. I wanted to look into the floral biology of the target species, wanted to try and determine the pollination vectors of the target species, and examine the breeding systems so that we could build up that image. I had 3 target species, Acronychia oblongifolia, Sarcomelicope simplicifolia, and Zieria granulata. So Acronychia and Sarcomelicope are both trees that are quite common within the rainforest communities. They're distributed up the entire east coast of New South Wales and into the southern portion of Queensland. The Zieria species I looked at, the Illawarra Zieria, is endemic to the Illawarra region. It's a rare and threatened shrub, and it's currently restricted to a few pockets of fringes of rainforest communities on that south coast. So you can see Sydney's up in the north, studying just that northern portion of the south coast for each of the plants. I was looking at in both remnant and regrowth rainforest community pockets for each of the plants, with the exception of Mount Annan. Oh, where's this cursor pointing? Let's see if I can do this up here. Mount Annan's up in the north, and Wollongong, which were both at the Botanic Gardens. And I would like to point out that for the Zieria, because that was rare and threatened, here's all the occurrence records, those small black dots. So the populations I studied were throughout that entire species natural distribution, just about. And there are less occurrences of it currently in the wild, as some of those records are quite old. So I'm not going to talk too in depth about the floral biology of each of the species, but in brief, I examined the pollen morphology, quantified the nectar production, looked into the floral morphology and phenology. For the pollen morphology, all the species were quite similar. So these are scanning electron micrographs that you're seeing. They get classed as small to medium sized pollen grains. And you'll notice they've all got that real heterobro shaped structure is what it's called, where it's quite undulate on that outer surface of the pollen grain. That's thought to be indicative of insect or biotic pollination vectors so that it will get caught on animal species. All flowers also produce nectar. It was in small quantities, but they're quite small flowers. So each of those images has been taken through a hand lens. So again, that's something that's thought to be a reward for animal pollination vectors and another hint that we're looking for biotic pollinators. In terms of the floral morphology and phenology, you can see each of the 4 species produce relatively similar looking small white flowers. And for the phenology assessment, so you I tracked a flower each day from its opening through to position or death. So Acronychia and Zieria were protandrous species. The pollen was always dispersed before a stigma became receptive, while Sarcomelicope was functionally dioecious. So the female and male flowers were on separate plants altogether for that species. So this is giving insights into the breeding systems as well. We're not expecting them to be self-compatible. And when we combine all 4 of these things, they also give hints, like I was hinting at as well, that these are what we call general entomophilous species, or best suited to that insect pollination. So I then wanted to look at exactly what the pollination vectors were for each of the species. And to do this, I had 4 different sampling methods that I undertook. Digital video recording, so I used GoPro cameras and I set these to record in the middle part of the day on hot days where insects were most likely to be active. I used time-lapse photography. These are the cameras you can see in the picture. So they were set to record from sunrise through to sunset. So we were getting that full suite of any cryptic species that might not be present in the middle part of the day when the GoPros were recording. I then undertook human observations. So these were done both diurnal and nocturnal, just in case there were some nocturnal insects or other nocturnal species that might have been transferring that pollen. And then nocturnal infrared motion sensor imaging was used. So these cameras were set from sunset through to sunrise, so that we were getting that full spectrum of every species. And when I say I did this, I had so many friends and family and volunteers that helped and sat and watched flowers with me and watched video recordings. So we ended up for each species with about 200 hours of diurnal sampling, 200 hours of nocturnal sampling. So to then identify each of the pollination vectors or the likely pollination vectors, we first had to identify all the floral visitors. So regardless of the method, a floral visitor was any species that came into contact with a flower during its foraging bout. And then a potential pollinator was any floral visitor that either carried pollen on its body or came into contact with the stigma when it was foraging. So looking at whether or not an insect carried pollen on its body, when we were doing the direct human observations, we captured insects after their foraging bout, euthanized them on ice, and then examined them under a dissecting microscope. This way we could ID them and see if there was pollen present on them. For species or individuals that there was pollen present, we looked at them then under an SEM, that scanning electron microscope, to confirm what species of pollen they were carrying and that it was that of the Rutaceae that we were looking at. So you can see in this third panel here, there's 7 Rutaceae pollen grains and 1 acacia pollen grain that has snuck onto that insect's body there. For insect species that were recorded during the GoPro analysis and the time-lapse photography, we noted whether its body came into contact with the stigma in its foraging bout, and then those were regarded as the likely pollinators for that survey method. So for all of the species, we got quite a diverse suite of insects that were regarded as floral visitors. There were a couple of flower spiders that snuck in and also 1 millipede that was crawling across an inflorescence in a nocturnal survey. But apart from that millipede, they were all diurnal species and all insect species, just about. So you can see the number of floral visitors were quite diverse, with over 70 species for Zieria, but about a third of insect visitors were potential pollinators. So most of them not acting as a good pollen vector. The potential pollinators came from a few different key families, predominantly the hymenopterans, a lot of Apis mellifera, but also native bees. You can see the two pictured. A number of dipterans, quite a lot of blowflies that were very abundant on the flowers for all three of the plant species. And then to a lesser extent, different coleopterans and lepidopterans, some butterflies and some beetles. For all of these species, they're common insects in rainforest communities, very common on the south coast of New South Wales. So it's not relying, or none of these Rutaceae plants relying on specialist interactions to have that seed set. We then wanted to examine whether some abiotic pollination vectors or wind could be transferring that pollen. So to do this, I set up a manipulative experiment in the lab, where I had an artificial wind source, a set distance from a flower. That flower was taped to a rod so that the anthers that contained visible pollen were exposed to the wind, and then a pollen trap was placed behind that. So that was just a petri dish with some Vaseline smeared on it. Wind speeds, we mimicked strong and light wind speeds in the area, so that was the 5 and 40 kilometres an hour. And then the pollen trap was moved either 5, 10 or 50 centimetres behind each open flower, so that we could try and capture the maximum distance that pollen would be transferred. So Acronychia and Zieria didn't have any pollen that was dislodged in any treatment. So even a strong wind gust or a storm, it's unlikely to move that pollen 5 centimetres and unlikely to be a pollination vector. For Sarcomelicope, pollen was dislodged in a few treatments. That's what you're looking at there, the Sarcomelicope pollen on some of those slides through a microscope. But this was really infrequent at light wind speeds, and it never moved more than 10 centimetres. And given that the Sarcomelicope is that dioecious plant, it's unlikely that in the field that it's acting as a pollination vector. So I then have the breeding system experiments. For these, there were 4 different treatment combinations that I applied. 2 were selfing, so the manipulative and the autonomous selfing. Both of these inflorescences were bagged with the polyorganza you can see in the image, so that they were excluding any natural or any insect visitors. The manipulative, we then hand pollinated with pollen from that same individual. Autonomous selfing, I left it and didn't manipulate beyond the bagging. For the manipulative outcross, they were also bagged to exclude any insect visitors or biotic visitors, but hand pollinated with pollen from a different tree of the same species or a different plant of the same species, but at the same population. And controls were marked and left open to natural pollination vectors. So to hand pollinate, the flowers were quite small. We had to view that there was visible pollen and then dab that onto a receptive stigma. We repeated this over 3 days just to maximize the likelihood that pollen was, or viable pollen was being transferred onto a compatible stigma. And then to determine the reproductive success of each plant, the percentage of flowers that fruited within each treatment was compared. Once the fruits ripened, we then did cut tests on the seed within each fruit. So we were comparing that viability again. You can see the seed on the left, that's white fleshy bit is the embryo sticking out. The endosperm is a small layer in there. It's quite hard to see in that image. Whereas a non-viable seed didn't contain either the embryo or endosperm, and it was empty. So that's that seed just there that's quite clearly got nothing inside it. For the results of this, for Acronychia, so on each of these graphs, I've got the treatments along the x-axis and the proportion of flowers that fruited along the y-axis. You'll notice whilst that's a percentage, this one stops at 20% because very few flowers set fruit in any treatment for Acronychia. So the manipulative outcross produced nearly 10% of the flowers that I manipulatively outcrossed set fruit, which was more than triple that of any other treatment. But they are self-compatible, it seems, with very few fruits set from that selfing treatment. And the controls where natural pollination vectors should have been transferring pollen also set very few fruits. We saw a similar trend with the Sarcomelocope. This time, about 40% of flowers did fruit when we manipulatively transferred that pollen. The control produced about half of that, with 20% of flowers producing fruit, and it's unlikely that they're self-compatible given we got no fruit from the selfing treatment. And for the Zieria, so this one was slightly different. There was no difference between the control and outcross treatment, so it seems like the pollination vectors are doing what they should for this species. They've still produced more than twice the amount of fruit from the selfing treatments, but again, this species, it seems, is self-compatible with nearly 10% of flowers fruiting from that selfing treatment. So then for the seed viability results, for the Acronychia, we did have some of the autonomous selfing treatment where it was left to see if it could reproduce without any pollination vector, producing viable seed. But this was very few fruits, and only 3% of the seed was viable in it. For the outcross and control seeds, about 10% of seed was non-viable. For Sarcomelicope, only a third of the seeds were viable, and we had close to 1,000 seeds that we looked at for this species. So something's happening where they're able to set fruit and then stalling that seed viability or not producing that viable seed. There was an extra category you might notice on this table, the partially filled. So what was happening here is we were seeing healthy and fleshy embryos within the seed, but there was no endosperm surrounding it. So ideally a germination experiment would be done to work out whether or not that's a viable seed. So yeah, nearly half the seed, we're not sure whether or not they were viable. And for the Zieria, while both the autonomous and manipulative selfing treatments produced fruit, not one seed was collected from any of those fruits. So likely self-aborting there. And then for the outcross and controls treatments, we had very few viable seeds that were collected. So the vast majority were non-viable, and this was largely due to predation, or in part, I should say, due to predation. So you can see over half the seed that we collected had signs of insect predation. So that could have been frost within the seed, an exit hole with an empty seed, or we did rear out one wasp that was fortunately coming out or emerging when I collected the seed. So these is signs of predispersal seed predation, and it was prevalent at all 5 sites that we studied the Zieria across its distribution. And we're fortunate enough to send this one away and get it ID'd. So it's a megastigmus wasp that's impacting this species. So in brief, shows that for the Acronychia, it's predominantly an outcrossing species and it's requiring a pollination vector and a generalist insect to set fruit. Similar for the Sarcomelicope, this is an obligate outcrossing species, which isn't a surprise from that floral biology where they're functionally dioecious, that's also requiring a generalist insect to set fruit. And the Zieria, again, it's predominantly or potentially an obligate outcrossing species, given we didn't get any viable seed from those selfing treatments and requiring that pollination vector. So when we look at the trends in this, for Acronychia and Sarcomelicope, Both species produced more fruit when the flowers were hand pollinated compared to when they were exposed to their natural pollinators. So they were pollen limited in that 2020 flowering season. But fruit production, when we analysed the results and took into account site and tree, did vary across and within the trees, across and within the sites for different treatments. So it's something else contributing to this, not just lacking pollination vectors. And a couple other theories that we have running at the minute are that it could be the environmental factors. We were in drought for the 2020 season, or other lacking resources that has led to that abortion of the fruit, or genetic factors. These are isolated remnant pockets of the rainforest, or regrowth rainforest that was only planted 20, 30 years ago. So potentially if the populations are already suffering that inbreeding depression, then that's contributing to this. So essentially that restates those 3 key points. And where was I going with that? But the outcrossing species requiring those pollination vectors, and brings back as well that point that we definitely need to restore the rainforest ecosystems, but potentially managing pollinator populations as well will have a huge impact for these species. So of course there's the acknowledgements. This project wouldn't have been possible without my 3 supervisors, Todd, Karen and Amy. I'm incredibly grateful for the time they gave me, as well as the countless institutions that let me work on their land, the entomologist who was so patient while I was trying to ID insects, and everybody else who helped sit and watch and bag plants. Cool, I think that's me done.